So, good morning, everybody, and welcome to the second Mistra SafeChem Breakfast Seminar. Today we have two talks. There's one talk that will be given by Per-Olof Syrén, and the next one, which is shared talk by Merck colleagues Lidia and Mojca. So I will give you a few instructions first on the setup of this breakfast seminar, and then followed by an introduction of the program before I leave the word to the speakers. So this webinar will run for 60 minutes. There will be 45 minutes presentations and then followed by QA session of 15 minutes. It will be recorded, and we will share the link both on LinkedIn and our homepage, Mistra SafeChem. And we will not share these presentations. And you're all invited to submit your questions using the chat, the QA, and then we will discuss those at the very end of this webinar. So warmly welcome. So my name is Patrik Andersson. I'm a professor in environmental chemistry and innovation coordinator at the IVL, Swedish Environmental Research Institute, working with sustainable chemistry and also leading this project, Mistra SafeChem. So today we have Per-Olof Syrén from KTH, the Royal Technical Institute of Technology in Stockholm, that will give a presentation, and then it will be followed by presentations from Merck, from Lidia and Mojca. Very looking forward to the talks. So firstly, a few minutes on the project, the Mistra SafeChem project. What is that? This is a project on green chemistry, where we try to have a holistic approach on developing of new chemistry, new molecules. So we work both with organic synthesis, catalysis, but also on hazard and safety assessment of these chemicals using different tools and different approaches in order to start good, so to say, starting knowing hazards and safety of the chemicals at an early point in the design phase. So this is a large consortium. It's led by the Swedish Environmental Research Institute. We are now in our second phase. We started 2020, and the second phase started last year, and they will run until 2028. The project is funded by Mistra, the Swedish funding agency, over these 8 years, and it's also nicely co-funded by industry, giving us 100 million Swedish krona per phase. All information, more information can be found on our homepage, mistrasafechem.se. So we are very honored to have a large number of distinguished partners, both research partners from two research institutes, IVL and RISE, but also universities, including the Danish Technical University in Copenhagen, KTH, Stockholm University, and also Uppsala University. And we have a large range of industry partners, and today you will be listening to Merck, but we also have AstraZeneca on board, BASF, Cambrex, Enginzyme, Lignicor, Norion, Pavstop, and Stockholm Vatten och Avfall. Great to have such a large and competent consortium. So the project is organized in a number of work packages, and we also have case studies. It's starting off from work package 4, where the new chemistry is being designed. We also work with materials, including textiles. Then we have 2 work packages working with hazard and sustainability. Work package 3, focusing on the hazard part, including in silico and vitro, but also chemical analysis. And then we have a work package 5, which is the life cycle assessment, taking a holistic approach on the development and on the life cycle of the chemicals and the products, the materials. All these key scientific work packages are collaborating in multidisciplinary case studies, the 5 mentioned here on this slide. And we want in this project to contribute with developing of the SSbD concept, the safe and sustainable by design concept that we are merging and talking about and discussing and collaborating in the work package 2. This was a very brief, short introduction of Mistra SafeChem. I will now leave the word to Per-Olof Syrén from KTH, Stockholm University. He will start talking about synthesis, and now I will stop sharing my slide.
Hello, good morning everyone. Can you hear me and can you see my slides?
I can hear you and I can see you.
Great, good morning. It's a pleasure to talk on this breakfast seminar. So I'm going to talk about a lot of green chemistry and synthesis, and actually then discuss a bit what is missing to make this really safe and sustainable by design at present. So what I do in my research, I combine enzyme and protein design with basically material science and organic synthesis. So we try to basically reinvent the way how molecules are made, functionalized, and also handled at end of life. And the goal is to take the whole process of such a molecular design and manufacturing into account. So more concrete what we actually do. So we are trying to basically develop using enzymes, mostly as catalysts, and we try to actually develop these fascinating guys into novel catalysts, making reactions that are actually up till now not observed in nature. And we can actually do that by revolution driven by generative AI, allowing us actually to create de novo designed proteins from scratch. And we are actually building automatized pipeline in my labs so that we actually can screen enzymatic reactions and also organic reactions in let's say larger throughput than what you can do if you do it manually, which is the traditional way. So we are really excited about this, and I will tell you a bit about some molecules that we have made, and one in this presentation I should add. And one of the things that of course is very interesting is the plastics problems. I mean, we are talking about 10 billion tons of synthetic polymer waste that has been accumulated since plastics was industrialized. So this is one of the challenges that actually my group is working from our let's say small perspective to try to contribute to, in actually making molecules and materials and also enable their end-of-life handling in more sustainable ways. Okay, so to give you a few examples of strategies that we work with here is like we can think about making, let's say, molecules from that are biosourced, right? So what we see here in the top is actually, you know, the idea, of course, using biomass. That is clear. It is very important. Now we can have a strategy where we use this biomass to replicate the existing molecules that are made in the fossil-based industry, such as, for example, synthetic polymers. But we are actually following another approach, which actually consists of using biomass and now not actually trying to force the biomass to become into petrochemistry, but actually trying to develop new materials that are more amenable to make from such biomass, resulting then in molecules that have a difference in structure, but ideally similar properties as the fossil-based materials. And we are trying to build enzymatic platforms for this job. And of course, now the key question is, actually to figure out whether or not this is possible and if it is also sustainable. So one of the key advantages actually of using enzymes in this respect is that they are selective. So it allows us, for example, from a soup of building blocks to selectively valorize them into certain molecules, only touching, for example, one type of bond or one type of molecule that is in this soup, for example, found in biomass. So this is a really high advantage. So we can make really nice functionalized molecules. And of course, now the idea here is to try to make them with green chemoenzymatic catalysis. So one of the classes of molecules that I think is personally quite interesting are the terpenes. So they are actually mostly cyclic molecules, and they are generated in all living organisms. So they are found, for example, as byproducts from the Swedish pulp and paper industry, and they are, I would say, underexplored in green chemistry. So we are trying to use this pool of biomass-derived building blocks and make interesting skeletons from them, so that we can generate molecules that actually replace fossil-based molecules, such as, for example, caprolactone that is shown here, coming from oil. So just to give you an example of some of the work that we have done, for example, we actually utilized chemoenzymatic catalysis to actually enable upcycling. You can maybe take this laser pointer so that you see a bit better. We wanted to make this kind of polymer, which would actually generate a structure that could replicate the properties of polycaprolactone. And then I predicted that we could use this guy here, alpha-pinene, to make this molecule. And this is now coming, like I said, from forestry. But to make this happen, we had to actually make, this is an inert molecule, so we had to oxidize it significantly, combined with reduction of this double bond that you see, to make it into this unprecedented cyclic ester. So we built a pipeline for this kind of green chemistry, which is shown here below. It was, as you see, from all the steps, all starting from biomass does not necessarily make things green. We can see some palladium chemistry here. We can also see some enzymatic transformations, which is very nice. But there are some questions appearing here about toxicity of intermediates generated, and also of actually the whole process. And although we can make this really nice new polyester, which has, cyclobutane scaffolded inside the polymer chain, which is very unusual, there are, of course, a lot of questions still about sustainability for such bio-sourced processes. Okay, so I will then show you some challenges. So we can also, we can make bio-based polymers with super interesting properties using terpenes as starting materials. And in the first example, which we published a few years ago, we could actually generate low molecular weight of this biosourced polyester. So this was actually challenging, limiting applications. We then, in the first phase of the Mistra project, we performed an oxidative cleavage of this pinene ring, so that we generated this diol here, which is shown. And now what we can do is we can make polycondensation from these two diol groups here. And now we can make polyesters by an alternative mechanism. And this led to much higher molecular weight, 7,000, allowing us actually to coat these materials generated onto different substrates. Okay. And now in this project, we're actually in the third phase of the material design, where we actually generated this terpene-based diols, which are super cool because they have a chiral handle on them. And now we can actually make polymers with, I would say, unlimited molecular weight. We also can show that these materials have very interesting properties in terms of self-healing. So when you break this material, it repairs itself. It's also working as some kind of super glue. So we are making our own post-its now in the lab with this material. But I would say that there are still a number of challenges. So another, cool, let's say, green chemistry possibility is to use also the plastics available as a carbon source to remake, monomers or building blocks from such plastic waste. So we developed also an approach that I call cascade depolymerization, which was recently published, where we actually start from plastic waste, and then we first do a chemical step, which is unselective to generate low molecular weight oligomers. So it's a pre-treatment where the degree of polymerization is reduced, the morphology of the material is affected. But then we apply our enzymes as a second step to actually then allow selectivity to be introduced so that we can take this soup generated in the first step and make some really useful molecules from it. So, for example, we developed actually a process for recycling of PET bottles where we use enzymes capable of degrading ester bonds. And then we combine that with let's say, microwave process to actually generate both a pre-treated version actually of the polyester, in this case PET bottle, and also allowing us to generate engineered enzymes that actually can act on such pre-treated material. So actually how this works is that we actually take a PET bottle shown here. So this is post-consumer. So our group was around the campus and collected actually some waste bottles. So we were called like the waste group for a while, which was quite fun. Then we took this bottle and we put it inside a microwave reactor. So it's not a normal kitchen microwave. It's a quite powerful one. And what happens then is that we can actually reduce the molecular weight of this polymer from a degree of polymerization from circa 100 to actually degree of polymerization of around 12. And what happens now is quite interesting. After microwave processing, we generate a very crystalline polymer. And we have now engineered enzymes that can bind to crystalline domains of PET. So we can put these two together, the engineered enzyme, the pre-treated PET bottle, allowing us actually to recycle a PET bottle at 30 degrees 1 hour in an enzymatic process, which is quite interesting. But now this is really cool, right? But what I didn't mention is that there are many things we don't know. What about the hazardness of the substrates, intermediates, etc.? What about transient byproducts or intermediates that are just there for a short time, but might significantly contribute to the toxicity of the process? And for example, in the plastic projects, we also have plasticizers that are embedded in the material. And they are sometimes not even considered in such recycling processes that I mentioned. We also have regioisomers. So for example, for the lactone case I mentioned, we can form different regioisomers. That is to say, isomers variants of the same product, and they might have different toxicity, which is often neglected. So I will just briefly mention in the last part of my talk what we have tried to do in terms of this, and really to highlight the need for better tools. So we looked on amide bond formation, which is typically happening under harsh conditions. It's done traditionally, for example, with harsh metal catalysis. So we wanted to try to generate a more, let's say, safe and sustainable process in this Mistra SafeChem project. The challenge here is that, I think you hear from me that I like to work with enzymes in chemistry, but the enzymes that do this kind of coupling reactions in nature, they are large and they are very difficult to engineer. So what we want is we actually want to have some kind of biocatalyst that is thermostable, it is active, and these enzymes that do these couplings, they are like mega enzymes, they're really like large, they have different domains that move, because the substrate has to move from one so-called carrier domain to the active site, which is located in another part of the enzyme. So we have to affect this motion somehow to deliver different substrates, which is extremely difficult. So actually, to do that, we actually developed a phylogenetic approach to protein engineering, which is actually was already delivered in the first phase of the Mistra project, where we actually can try to recapitulate the evolutionary history of proteins to generate an ancestral variant of a certain existing enzyme. And the idea here is kind of use existing sequence information and statistical bioinformatics to enable us to travel back, millions or even billions of years ago, where this particular enzyme perhaps was stable and perhaps did some really interesting chemistries. And we have shown that it's possible to actually transform this into a platform technology. So we have many examples of where we actually can use this technology to actually affect, let's say, how this enzyme or protein is moving around, how it's wobbling around, how the motion is. So I will not go into the examples, but we are talking about, introducing 50% change perhaps in this catalyst. We are talking about hundreds of mutations that are being introduced simultaneously in a gene by design. And it's remarkable to see that we can still generate active enzymes using this approach. So then the idea was actually to, in collaboration with AstraZeneca in the project, to look into these so-called ABS enzymes that make this coupling. They have two domains, the N-terminal domain and the C-terminal domain. So the first part of the reaction is the carboxylic acid couples to the energy currency in the cell, the so-called ATP molecule, and forms this adenylated intermediate. And then in the next step, this domain has to rotate circa 90 degrees compared to its original state, so that this amine substrate can now bind here to this rotated state, the so-called amidation conformation, so that the click can happen, the reaction occurs. So the idea was to use our approach in the Mistra SafeChem project to try to affect this motion of the domains, to in that way make some kind of super enzyme that can actually accept different amine substrates. So this is a lot of scientific details. I will not have time to go through into all the details, but remarkably we can show that this works. So we used our approach and generated an old variant of this existing ABS enzymes. And what we found in this ancestral enzyme, which is represented by this primitive human being here, is that actually this C-terminal domain is more flexible in this ancestral variant compared to the extant counterpart. And why is this? Well, we can go into the molecular level. Of course, I'm a scientist, so this is what I love to do. And we can see that in the modern enzyme, the existing guy out there, a lot of hydrogen bonding and electrostatics keep these two domains tethered together, making them kind of rigid. But now when we make our variant enzymes, we are introducing 186 mutations. We can see that we break these interactions. And this is why we think that this domain now becomes more flexible. And what we then can do is we can then take, of course, in a very nice collaboration with AstraZeneca, we can then take the catalyst that we develop and we can actually make something useful. So we can scale this up to preparative scale, actually making some very interesting pharmacophore molecules, which is highlighted here. So this was just published a couple of days ago, this paper. So I have a few minutes left, so I will tell a little bit how far we reached and what is left to do in the safe and sustainable approach. Because what I didn't mention is all of these things. What about, toxicity? What about safety of a worker in the chemical factory that is handling these substances? So in what we did in the amide case, which was a super nice collaboration with IVL, RISE, Stockholm University, and also AstraZeneca, is DTU, I should mention also. We had a super nice collaboration here where we actually developed a pipeline, an in silico pipeline, allowing us to predict the toxicity of building blocks before the process is designed. So we generated using basically the Mistra SafeChem toolbox. To simplify, I don't really have time to go into the scientific details. It's an AI, driven method which is taking, receptor binding to human receptors, persistence prediction, what happens to these molecules in the environment, etc., into account. So we built a platform around that, allowing us to pull in a smile string into this filtering mechanism, and then what is coming out is some kind of prediction whether or not this molecule is safe. So we pooled about 100,000 molecules through this in silico filtering, and then we came out actually with a panel of building blocks suitable for amide coupling that were predicted to be safe and sustainable by design. And I call these molecules the SafeChems. they are safe and sustainable by design before the process is even made. What we then did together with AstraZeneca is we took all of these SafeChems and we combined them together using our engineered enzymes. And this is a lot of results from the lab. We're talking about significant amount of coupling chemistries here. But what we see is that we can generate with our variants really interesting molecules. And I mentioned this variant that we generated. We can see that we now actually have a super enzyme that is accepting, that has a broad amine acceptance. So it's actually capable of taking these building blocks and making very nice amide products from them. Okay, so this is results from DTU, which is very nice collaboration where they used actually the use tox evaluation to try to predict, actually to verify the in silico filtering approach I just talked about. And we can learn a couple of important things from here. Again, I don't really have time to go into all the nitty-gritty details, but what we observed is that typically in this case for the amides were predicted to be more toxic than the starting material, meaning that we can not only use the starting material as a proxy to predict actually the toxicity of the final product. We can also see a little bit where these molecules cluster in the chemical space. So this is again emphasizing, I would say, actually the need for actually developing better tools to predict the toxicity of actually these kind of molecules. Because let me take this picture and show you where this ends up in the global chemical landscape of all chemicals that have been analyzed. And this is like, again, I really want to thank Kerstin here. She's great to work with in this project. And what we actually can see is that for our amide guys and the responding starting substrates, we can see that the training data that was used to predict toxicity, they are far away from where our molecules cluster. So that means that there is a gap of the training data that has been used to train this model and the actual chemicals that we used in the Mistra SafeChem project. And this can be seen here in my last slide, which is a collaboration with Park in a manuscript status, where we actually can see that most of the compounds that we worked with in the amide case were actually predicted as non-toxic. But we know that some of them are toxic because they are aromatic structures, etc. So with this, I will actually conclude, and I will say that we need better models. And really, this is super important. And I think this we will hear more from in the next presentation, where I think this will be mentioned more in detail. So we are on the way, but better models are needed. So with that, I will thank you so much, and I'll stop there because I think I'm running out of time probably.
Okay, so hello everyone. I think now is our turn. Let me share. I hope you can hear me and see me all well.
Now it's perfect.
Thank you. So it is really a pleasure to present in this edition of the Mistra SafeChem Breakfast. Today, myself and my colleague Lidia will be sharing how we at Merck are leveraging in silico new approach methods to support the application of a safe and sustainable by design framework. Lidia and myself are part of the Merck Life Science business in the Global Regulatory and Computational Toxicology team under the umbrella of Life Science Quality and Regulatory Management. Merck Life Science brings to the biotech and researchers in 66 countries across the globe a complex portfolio of over 300,000 products. This really highlights the needs for solutions at scale, where really the speed and confidence in hazard identification, as already mentioned before, really matters most. And this needs and such a complex portfolio gives us a lot of opportunities for such at-scale solutions, but also a lot of challenges. So let's begin. I will be sharing on the in silico approaches and how we develop and apply those approaches. As mentioned, we recognized a strong need for the hazard identification. And let me start by saying that it is really now the time to move away from traditional animal models to evaluate the hazards of the chemicals into new approach methods. And why? Because the new approach methods are offering us the possibility to have safety evaluations that are more human relevant. This strong commitment to move away from traditional testing is observed among the scientists, among industry, and also among the regulators. In fact, in the Q1 2026, we are expecting the European Commission to publish the roadmap towards phasing out animal testing. And as mentioned, the hazard identification is needed. We need to speed it up. Why? Because we have to classify and label our chemicals to inform safety data sheets. We need to register the chemicals with authorities. And by giving new approach methods a chance in our application, we are really contributing into the reduction on the reliance on animal testing, as well as through such approaches, we are also promoting a toxic-free environment because chemicals have more robust hazard identification information. We have mentioned new approach methods. So let me share that for our internal scope, we have adopted the definition of the new approach methods as is proposed by US Interagency Coordinating Committee on the Validation of Alternative Methods, where it is really emphasized that new approach methods are any technology, methodology, approach, or combination thereof that provide information on the chemical hazard and risk assessment and supports the replacement, reduction, or refinement of animal use. And let me also add how Worth and Berggren nicely put it, that there is no longer the challenge in the lack of methods, but it's more that we have to make those methods practically available and acceptable by regulators. So now that we understood what really new approaches comprise of, we can appreciate that there is a vast arena of such methods. And for our internal purposes, so we are currently operationalizing and leveraging computational modeling and grouping. So let's now understand what are our main components of our solution. And let me start by saying that these are the methods, in silico methods, the data, and the key point, the experts and their expertise. As I have mentioned before, we have very strong drivers to develop such solution: the complexity of the portfolio, the confidence in hazard identification, the speed and scalability, and of course, the regulatory needs. So within our solution, we are maximizing the use of existing knowledge by defining the ways of assess and use regulatory data. We are also qualifying existing data in this new context of uses. Then we have the QSAR predictions across all relevant endpoints provided by the QSAR models that are compliant with regulatory requirements for such models. And by unlocking really the added value of the existing data and the predictions and the data that is coming out of the QSAR models and grouping in the very, let's say, robust, expert-driven ways of evidence approaches, we are really able to deliver data timely so that also new chemicals or chemicals that have data-poor substances can have robust assessments. But as it was already presented in the previous presentation, we are aware that no one solution fits all. And that is why we are also strongly motivated to collaborate under the Mistra SafeChem program. In fact, we really appreciate that this program brings together multidisciplinary teams covering researchers arena as well as industry, where we can discuss different applications, different uses, different needs, and leverage really from this, let's say, strong background of every expert. As mentioned before, it's a key component of such alternative solutions. Now let's see what is the current solution that we are using in Merck. The building blocks that I have shared with you before were used to build a centralized computational platform. Within this platform, the endpoint-specific workflows are automatically executed to the extent possible. And for these workflows, as anticipated, we have built strong and robust expert-driven criteria for evidence integration as well as for reliability assessment. What I would like to stress is really that these workflows are mimicking the way an expert would use data and would use the models. These are not simply automatically executed. So this is really, let's say, an expert supervised approach. And additionally, because we want our solution to be adaptable to the current regulatory needs, to the forthcoming regulatory needs for hazard identification, each building block, prior its integration into this our QSAR application, it is interrogated against some criteria that we really find critical for our solution. And this is being relevance, transparency, reproducibility, and scalability. As I mentioned before, we are really motivated to collaborate and also within the Mistra SafeChem program. We have joined Mistra SafeChem program in its second phase, so now it's over 1 year. And as already mentioned in previous presentation, within this program, an in silico toolbox has been developed that comprise of several QSAR models across key relevant endpoints touching upon human toxicity and environmental toxicity. In this past year, our team has really focused onto having a deep understanding of the QSAR models that are implemented into this tool, to understand it from our perspective. What are the benefits for us? What challenges we are seeing? And how is there any opportunity for improvement? Can we provide some guidance on the application of such tools? And also, what is very relevant for us, if we want to incorporate this solution into our systems, we have to have a clear picture of what it is the compatibility that it has to fit for our internal purposes. And as mentioned, through collaborations, under this program, we want to advance and leverage Mistra SafeChem in silico toolbox optimally. We want to develop new models to inform hazard identification, and we would also like to test the applicability of artificial intelligence for toxicity prediction. So our ultimate goal is really to have a deep understanding of the modeled endpoints in the context of their regulatory applicability, its context of use. We want to have a user-friendly solution that is adaptable to our internal solution. And this all to finally come to a point where we can confidently integrate the models that have been developed under Mistra SafeChem to run the predictions and assess the hazards that is then can be adapted also for regulatory submissions. In this way, we can expand endpoints and enhance our predictive capacity. With that, I will move to my last slide to summarize why all this matters. As anticipated, we can appreciate that in silico, new approach methods, in fact, are enabling faster and scalable hazard identification that is aligned with refine, replace, and reduce principles, as well as safe and sustainable by design goals. Currently, under the program, we understand the context of use of the current status of the in silico toolbox, and we have the tool currently running as a standalone tool on our premises. What we would like to achieve, we would of course like to advance the toolbox and have regulatory context applications. We would like to develop new models for aquatic, targeting specifically chronic toxicity. And of course, we would like to further support the development of case study under the SSbD program. And with that, I'm happy to pass over to my colleague, Lidia.
So thank you very much, Mojca. Good morning, everyone. So now I'll be presenting you the SSbD case study that we are conducting under the Mistra SafeChem program. It's focused on solvents, and we started only this year, so we don't have yet results to share today, unfortunately. And therefore, we'll be focusing more on how we are proceeding with this collaboration and also on the key steps that we've taken so far and also that we are planning next. Next slide, please. So in this case study, we are comparing the safety and the susceptibility profile of NMP, N-methyl-2-pyrrolidinone, which is a widely used important solvent, but is also developmental toxicant. So there's an urgent need to replace it with another less hazardous solvent. In this case study, we identified 4 alternatives, and we are focusing on the use of the chemical synthesis in the lab, because as you know, solvents have got many wide uses, but we had to narrow down a little bit the scope. We are collaborating with IVL, RISE, and DTU, and we created different sub-teams. So one team is dedicated in the hazard assessment, another one on the risk assessment, a third one on the environmental LCA, and a fourth on the social LCA. We started to work on this case study in May, and we are planning to finalize it next year. We're using also a plethora of tools. Here we reported some of them, as Mojca already mentioned, the Mistra SafeChem QSAR toolbox, but also Merck's QSAR app, for the risk assessment CESAR, and eventually also higher tier risk assessment tools. For the LCA, we're going to use EcoInvent and also an LCA modeling tool. And for filling data gaps in the LCA data inventory, also Cynthia, it's a retrosynthesis tool, but also in-house process simulation Excel calculator that IVL developed. In addition, also an external AI tool that we are using in Merck. Next slide, please. So in this slide, I report an overview of the solvents and assessment. For confidentiality reasons, we have input the names and the structures, and these solvents, however, been proposed to us by colleagues working in the R&D department, and also some of them are quite known in the literature. All these solvents have got a rich registration dossier, and we also reported in purple the registration estimated tonnage band as available in the ECAChem website. And as you can see, only NMP bears this harsh classification, so this confirmed developmental toxicant as a classification, whereas the others eventually acute oral toxicity, skin irritation, or no hazardous classification at all. Only one solvent is still under investigation for potential reproductive toxicant effects. So there's our OECD 443 yogurt study that is going on, and the results will be ready next year. However, the trigger for this assessment is the hazard concern of NMP. Next slide, please. So the key objectives of this case study for sure is to identify the most suitable solvent, but in this program we also want to test and evaluate new and existing tools and also identify the best practices for addressing the data gaps, because of course we will be always having data gaps. For the hazard assessment, we also want to identify areas of improvements in the Mistra SafeChem phase 1 QSA models, as Mojca already anticipated, but in addition we would like also to plug in more mechanistic toxicity evidence. And I will better explain it in the next slide. For the risk assessment, we want to assess the suitability of also conducting a risk screening analysis using the ProScale tool. And also for this part, I've got a dedicated slide. For the LCA, as you know, companies have got many data gaps in their data inventory, so we want to evaluate and choose different tools in order to fill these data gaps. For the social LCA, this is something that it's quite new to our team, and we are really glad that we've got in IVL and RISE social LCA experts. So now we are working together in the identification of the most relevant social indicators, and also we needed to find ways of filling data gaps. And something that is coming up, also together with them, we will be developing a practical tool for running social LCAs. So more to come in this regard. As you noted, perhaps I haven't mentioned the economic part. This because also social and the social economic sustainability assessments are quite new to our team. So we want to tackle first the social, and then we will try to better understand how to tackle the economic evaluation. Next slide, please. Okay, for these solvents, we are quite lucky because they've all got rich doses. So we've got many hazard data, and we immediately started to compare the experimental data that we collected from the rich doses with the QSA predictions that we obtained from Merck's QSA RAP, but also from the Mistra SafeChem QSA toolbox. We wanted to check a little bit the concordance of the data, but at the same time also assess, of course, the reliability of the predictions. And from this initial assessment, we identified the next steps. So first of all, we identified the endpoints that required further investigation with a literature search, because either we achieve ambiguous predictions or because we were lacking experimental data. In this case, we are talking about endocrine disruption and carcinogenicity. Then we would like also to evaluate a new computational toxicology tool that we just acquired in Merck, which is based on omics data. And this tool is able to predict the activation of metabolic pathways. Why we want also to include it in this case study? Because we feel the need of adding mechanistic evidence. So as you know, for IAT endpoints like carcinogenicity, developmental toxicity, in this case, if you don't have experimental data, you cannot make conclusions based on QSA predictions. So there's a need to shift to mechanistic toxicity evaluations. We would like also to define which NMP developmental toxicity mechanisms are predictable from the collected data evidence and assess the applicability to other solvents. Afterwards, based on this evaluation, we will eventually revise the hazard classification and data and run the risk assessment. Next slide, please. Okay, so for the environmental LCA, so far we identified the goal and scope. So it's a cradle-to-grave assessment. Therefore, we are going to consider the whole product life cycle. And as a reference unit of comparison, we are going to assess 1 liter of solvents for the chemical synthesis in the lab. Currently, my colleagues, and together with IVL and RISE, they are working on the data collection piece. So they are collecting data for filling the data gaps in the LCA data inventory, using literature data, EcoInvent, an external AI solution, IVL in-house process simulation Excel tool that perhaps also will be refined, retrosynthesis tools like Cynthia, that is a tool owned by Merck, but also IBM RxM. Once they will finalize this data collection, they will run the LCA model and assess the results. For the social life cycle assessment, we're really at the initial stages. So now we are working on the identification of the relevant social indicators, so for solvents, but also for chemicals. Could be indicators that are relevant to the human health and the safety, or to community impacts, working conditions. So we need still to choose them. And then we need to see if we've got data internally in Merck and at product level. And if it's not, how to fill these data gaps using these databases reported in this slide. So the work, as you noted, is still ongoing. Next slide, please. So ProScale, it's a tool that has been developed already some years ago in industrial consortium with experts coming from both the risk assessment and the LCA arena, including also IVL experts. This tool uses a scoring system to measure hazards and exposure. So with ProScale, we are able to assess the potential of the chemical exposure to workers, professions, and consumers throughout the product's life cycle. I like to call it that is an hybrid tool because ProScale is like the LCA tools, uses the concept of functional unit and mass flow, which are not concepts that are common to the risk assessment community. But like the risk assessment, it uses hazard data in tier 1 ECTOC tier A exposure modeling to estimate the human health risk potential. So in this case study, we will be evaluating ProScale to understand if we can use it both as an early risk screening tool, but also for the calculation of the human health toxicity impacts first and then ecotoxicological impacts afterwards in the LCA calculations. So testing is ongoing. Next slide, please. So in conclusion, this SSbD case study on solvents is still ongoing. We are planning to finalize it next year. It's a collaborative effort among industry research institutes. The goal is for sure to test and evaluate new and existing tools, but also to establish best practices to address data gaps. Once we finalize this case study on solvents, we would like to start another one, most likely in 2027, focusing on different product types and research questions. And with this, I would like to thank you for your attention and we're happy to take any questions. I will turn it over to you, Patrik. Thank you.
Thank you so much, all of you. Great presentations and very good, nice overlap in between the two presentations, or nice combination with your developing of new chemistry, Per-Olof, and the developing of the tools. I don't have any questions from in the QA at the moment, but I just would like to address to both of you, or the three of you, a few just comments, and starting off with Per-Olof, you ended up your talk about challenges in the modeling of your key components in your amine chemistry. How do you see that we can move forward in the society here, and also, of course, in particular in the Mistra SafeChem project to improve model output for this particular chemistry? You mentioned the low reliance, the lower scores of this chemistry. What do you think are the major challenges there?
Yeah, that's a very good thing. Can you hear me?
Yes.
Super, because it said that my mic was muted. Okay, cool. So yeah, very good question. So I think we have several challenges. So one is that the particular substances that we have been working with, which are aromatic based carboxylic acids and amines, they have not been well represented in the training data set for the models that we have been used for in silico filtering and tox prediction. So there is a gap identified in what the training data, what that was plugged into the models, and what we actually are in need of for our particular chemistries. So this is a challenge that can be solved, of course, if we now can train these models with compounds that are more representative of the ones that we are using. That's one thing, and the second, so that I think I'm quite optimistic about that. Then we have also, of course, a challenge that I just briefly mentioned, which is when you perform these reactions, you sometimes have this very, let's say, low concentration, highly transient species that might exist, in your reaction mixture, but you might not even observe them, and they might significantly contribute, to actually the sustainability of the process, and especially in terms of toxicology. They might even, persist into the product even. So and we might not even see them. So that is also a challenge. So that can be solved, of course, with with high resolution mass spec. But I see that those are challenges that somehow we need to address in the project. And I think we can do that because we have the right. It's a very nice collaboration. So I'm quite optimistic.
Thank you so much. I think so. What you stress is we need more experimental data in certain chemical domains to improve the models, and I think that's absolutely something that we urge for also in the project. And we need to collaborate to enrich our data sets for model improvements. Even though artificial intelligence is getting into the field and the models getting better, we also need data for training, obviously. I think also it's interesting that you mentioned transformation products, because there's a lot of things going on, both on how we predict the transformation product, but also on the challenges in predicting effects of transformation products. May they be abiotic or biotic transformation products.
Yeah,
still is moving there. So thank you so much for a very interesting talk. Per-Olof, and I now turn to Mojca and Lidia. You are obviously working with an enormous chemical domain, so lots of chemistry in the portfolio of Merck. What do you see of challenges there in moving away from animal testing to predictive tools in your domain and in your portfolio? Are there any particular areas that you would like to stress?
Maybe if I can give it a go. So yes, the challenge of our portfolio is its vastness, and low volume chemicals, which it means that by regulatory requirements, the request to have in the past, let's say, to have data for toxicological assessment was not really there. So how to characterize all those low volumes where it is a challenge. So that is why we see that we definitely need to leverage the new approach methods to its maximum, I say, to really maximize it. This and then not only by applying the new methods, but also look what we have gathered in the past on similar compounds, so that we can group our chemicals to start getting insights based on what is available in the literature, based on what is available on similar compounds, maybe to support us in then selecting chemicals that are, let's say, representative of groups where we can then test. Then, based on the knowledge that we can get from the current data, from the current models, to drive and design specific studies. That's why really the new approaches, and as Lidia mentioned, the going further into the mechanistic domain. It is really important to leverage this, and integrate into our daily operations, because we need the hazard to be identified. Now, because of a flood of low volume chemicals, there is a request to have hazard identification also for those, to know that they are sustainable ultimately. So now we are moving and facing and along these lines, how to tackle these low volumes where really data are scarce.
And if I may add, we are also trying indeed to apply what Mojca introduced in our case studies. So this is really a good opportunity also under this program to test new tools, to play a little bit more with the, with the data and the tools, so that we can come out with new evidence and also new ways of combining these methodologies, so that we can make informed decisions in the end of the road.
Thank you so much. I think this is a great opportunity to, so to say, stress test tools and find the gaps and find the challenges and move towards solutions where we have intercollaboration on board in this particular project. This webinar is going towards the end. I would like to thank all three speakers for excellent talks. I would also like to stress that we do have upcoming breakfast seminars. There will be organized new seminars during the spring and the autumn of 2026. And if you have comments or questions after this presentation, you are of course welcome to approach us, me, Patrik Andersson at IVL. So thank you so much and have a good day. Goodbye.
Thank you very much.
Thank you. Bye bye, everyone.
Bye, everyone.